direction in cancer treatment, one that moves past the limitations of traditional chemotherapy and opens the door to safer and more precise generation of immunotherapies. It's an emerging approach with major scientific promise and significant implications for global biotech investment. This keynote is titled as Beyond Chemotherapy, PLPC-DB as a new de-risked non-cellular immunotherapy asset class in global biotech's investment. Allow me to please welcome with a round of applause Dr. Ramón Sandoval, Chief Scientific Officer at OGRD Alliance. Are you here? Yes. A round of applause for our next keynote speaker. The stage is yours. Check. Testing. Test 1, 2, 3, check.
Hello, hello, hello. Here? Testing. Okay.
Check test. 1, 2, 3, check.
Okay, let's go. I need the first. Okay. Thanks for your attention. Today I present PLPC platform. But first of all, what is the scope? What is our scientific pipeline in 2025? In this year, we have 4 works presented in ASCO, American Society of Clinical Oncology, and 2 works in ISMO, European Society of Medical Oncology, and 3 works in Society of Inmunoterapia in Cáncer, San Diego, California, in March, and 3 works in Orlando, September, in CAP25, College of American Pathology, 5 papers, Q1 peer-reviewed, published, 4 books available in Amazon, 3 family patents present across 3 continents these years, and 2 external validated audits from Fryer and Veristat about to pre-FDA access. But what is PLPC? PLPC is a platform with 3 modules. We have PLPC-DB, dendritic-based, that is DB, personalized cancer immunotherapy, NX, vertical, 4 lines. STIP is our backbone regulatory. In PLPC NX, we have 2 areas. There are support for T defense and Regenera, and wellness for Vitalis and sport. How works PLPC? PLPC uses a new language. If you see the immune system, some therapies get believed as you can treatment with a cell or with a molecule, but in reality, the difference is that the immune system is all new language. For me, excuse me? Okay, perfect. Thank you. Okay, more? Thank you. For many years, the immune system, so body could only attack or defend, you know? But into the immune system, use multiple molecules and signals to correct and reprogram the without damage. Immunotherapy has evolved throughout a century, from Dr. Virchow to Steinman, and we have many papers and books, thousands in reality, but very few really treatments to the patients. PLPC is not a concept, it's not a startup, it's the apex of the entire. Testing. Perfect. This is better. So immunotherapy from Dr. Virchow to Steinman evolved throughout the century. And PLPC is not a trend, not a concept, not a startup, not a new project, not an idea. It's the apex of the entire journey. It's a mature synthesis, you know. The Nobel Prize. In 2011, the Nobel Prize goes to Dr. Steinman, Butler, and Hoffman, because they showed how the immune system really works, what to attack and what to tolerate. And they discovered the adaptive immunity, you know, the getaway of tolerant receptors and dendritic cells, starting from our skin. This is a new logic. PLPC represents a new logic. For decades, we tried to replace the cell, CAR-T, or inhibit them, checkpoint inhibitors, anti-PD-1, anti-CTLA-4, for example, or force them, many cytokines, EL2, EL10, anyway. But they all share the same weakness, depending on unpredictable living biology. And this is the problem with CAR-T, this is the problem with dendritic cell, this is the problem with stem cell therapies. So PLPC is different because it's a structural therapy, not dependent of living biology. Later we see about structural immunotherapy. The skin. The skin is the gateway, is where the immune response starts. You know, let me see. In the skin, you can see the dendritic cells, a special kind of these cells, Langerhans cells. And this Langerhans cell or dendritic cell get the message, sense what happened on the superface, and translate the message to the immune system in the deep layers. So PLPC uses the same root of this natural message. PLPC follows speaking the signaling language of the dendritic cell with a minimal intradermal application. The PLPC works with an antigen presenting system. In this system, the dendritic cells get the antigen and put some parts in the surface, and these parts connect with helper T cell that allows connect with killer T cells. And killer T cells destroy cancer cells, infectious bacteria, virus, anyway. So the PLPC follow the function about dendritic cell and T cell because we brings harmony to the immune system with Th17, Th1, with for defense, with balance, Th2 for cooperation, and with Tregs. So with that way, we get a given functional equilibrium. What happens if we combine molecules or signal from DBT, dendritic-based therapy, with conventional therapies in cancer, chemotherapy, radiotherapy, checkpoint inhibitors? What happens? For example, many studies show us that when you combine chemotherapy with dendritic-based therapy, chemotherapy efficacy rise up to 50%, and side effects fall 40%. So anorexia, for example, 53 combined therapy versus 96. Leukopenia, 43 versus 80%. Abnormal liver function, 41% versus 70%. This is a big difference. And how is possible? Because PLPC supports 5 mechanisms. First, antigen presentation, so increase tumor recognition. Co-stimulation with signal CD40, 80, 86, so increase immune efficiency. Increase synergy with chemotherapy through Th1 cytokine release. Treg modulation, restore anti-tumor immunity, and immune memory, so decrease the recurrence. 5 mechanisms to improve the efficacy of chemotherapy. Radiotherapy is a local treatment, but under DBT, under PLPC, we have abscopal effect. And how is that? Dendritic cells capture antigens when local radiation released from the tumoral lesions. And these T cells activate CD8 in the lymph node, present these molecules to an attack distant tumors. So this is the way that local therapy becomes a systemic immune response. All knows that the cancer builds its own ecosystem, silencing lymphocytes, deprogramming defense. So PLPC is a restored functional dialogue. If you see, if you see, the immune system have multiple molecules and cells. So when we use checkpoint inhibitors, one molecule, or CAR-T, one cell, we fight against all the entire platform. For that reason, if you use checkpoint, you can repair or use one key. But if you use a platform like PLPC, you can reset the entire cable. You know, this is the difference. For example, you can see the curve response, usual in the checkpoint inhibitors. In the green bar, in the third graphic, you can see the difference when you use DBT, dendritic-based therapy, like a warm-up, some weeks before the treatment with checkpoint inhibitors. And in the last graphic, you can see the effect when you use at the same time dendritic-based therapy with checkpoint inhibitors. So you can get higher efficacy and longer-lasting outcomes over time. Quick comparison. Drugs, a strong response but frequent adverse events. Exosomes, weak response with few adverse events. And PLPC, a strong, durable response with minimal adverse events. The main profile is zero toxicity. For that reason, this kind of immunotherapy is for all stage in cancer, for each type of cancer is personalized with no systemic toxicity and synergized with any therapy of conventional treatment. This is how we move from chemo units or surgery rooms or radiotherapy units to the safe, minimal, at-home application. You don't need the environment of hospitalization, even without leukapheresis or intravenous immunotherapy, that the older kind of immunotherapies in cancer. Remember that concept? This is very, very famous. Multiple tools in just in one device. Few believe it than the beginning. PLPC follows the same route. Many benefits in just one therapy. Release cytokines in order, like chemotherapy, synergist, radiotherapy with ascopal effect, and surgery, removal, residual implant, thanks to the immune memory. This is the whole sequence. Very simple, the show. We get a liquid of cells, PBMC, peripheral blood mononuclear cells, and we generate a cell culture enriched by tumor neoantigens. So in this step, we get the secretome, and the secretome we proceed to centrifugation, concentration, lyophilization, and finally administration application intradermal, minimally invasive. So this is simple, but behind that slide, maybe 240 protocols, and each protocol with 30 or 40 drafts before. So this is not a rocket, this is entire NASA, you know. This is the behind. And let's see the tumor antigen, because yesterday after the panel I received some questions. This is the special step, tumor neoantigen, because if we replace tumor neoantigen for example, senescence molecules, we can get immunotherapy for anti-aging purpose. If we replace tumor antigens for a special profile of cytokines, we can get a tolerogenic immunotherapy for autoimmune disease, for example, to transplant, reject to transplant. And that is not an unpredictable, not is random biology, because we have very consistent markers, CD25, 40, 69, 86, IL-10, in our profile of cytokines and interleukins, and our proteomic spectra reveal multiple protein platform, main 5 blocks of 20 protein each. So it's a reproducible, very precise biological identity. From the real-world evidence, after more than 3,000 patients across 11 years, we have many data to process. But for example, PAT with 18 FDG, up to 80% reduction in metabolic activity after 45, 60 days approximately, with zero systemic toxicity. This is the how it goes on after. What is the scope to the registration in some agencies, regulatory agencies? For example, in USA, FDA expanded access 3 months. Mohab, 3 months, compassion use. And Singapore, HSA, for special access route, under 3 months. But this is immediately registered, because if you get a formal, finally, registration, more than 12 and 34 months, because it's a new way, not the 10 years, not the $500 million, like a normal or conventional drug, because use the special way. We can see later. The real-world evidence and plus the STIP provide us animal-free stage, phase 1 to 3 free. Later we see how it's possible, and lower cost and shorter timeline to registration. For that reason, many people consider that PLPC platform or other like that, there is very non-pharmacodynamic treatment at this moment, like an advanced secretome platform, maybe the first in class. This is a quick summary. Best entry value, zero toxicity, lower risk, and the better stability profile score to the entry. The second pillar of this platform is STIP. STIP is a structural traceability and immunophenotypic platform. It's a regulatory backbone. It's that make the magician is possible. You know, STIP allows us human ex vivo evidence engine, also CTD 5.3 documents generator. All of that, NAM, new approach methodology, aligned and compatible with Part 11 digital traceability. For that reason, STIP allows us formally eliminate animal studies. This is from FDI Act Modernization 2.0 from 2022. And the new approach methodologies does not bypass phase 1 to 3. Replace them, but with equivalent data, but faster, safer, cheaper, and more accurate. It's a new way to get evidence. STIP belongs to the third type of the NID FDA, ex vivo models. Other type of new approach methodologies are organoids, organoids-on-chip, and silico simulation. So in that way, STIP get data that converge in module 5.3 for biomarkers and traceability integration. This is the way that the bridge between science and approval. But the STIP is not optional for the PLPC. It's the only way path, you know, for non-pharmacodynamic treatment, because PLPC has no PK/PD, no dose response curve, no clearance kinetic, And it's structural, not pharmacological. You don't have a systemic circulation or a plasmatic concentration or levels. You don't have this because it's structural. You apply intradermal molecules, and these molecules is absorbed, assimilate with the membrane cell in the cells, and start the new work, the new terrain to the chemotherapy, radiotherapy are really functional. The timeline with STIP is more shorter. In the right side, you can see the difference between traditional path but STIP pathway. How is shorter the timeline. Animal studies 1-2 years replace it, almost instant. Phase 1 more than 40 hours because it's a study with different lines. Okay, perfect. And phase 2 1 to 2 years. Phase 3 3 and 7 years is replaced by matrix, so it's more shorter. The applications of TIP in the industry is multiple. Biopharma, phase equivalent validation, accelerated non-pharmacodynamic pathways for nutraceutical, get first a mechanistic proof of function, diagnosis, structural biomarker validation for CROs, traceable ex vivo validation, and sovereign agency, and modernization of the regulatory programs. This is very, very interesting because STIP get more than 20 parameters are similar with Nash game theory. Analysis unit, traceability, convergence, input, function, and centralization, and 25 more. This is a flash, better evaluation, shorter timelines, faster process, regulatory cost savings, and fewer regulatory compliance to any jurisdiction across the world. And finally, the NX. NX is 4 lines, T-Defense, Regeneron, Vitalis, and Sport. The main characteristic is only requires notification because all components is fully GRAS certified, so don't need studies for any molecule or any component. We work with 6 axes, and with this axis of composition, we get different purpose, different benefits for different markets. You know, Regeneron is for autoimmune and inflammatory chronic disease, Vitalis for anti-aging or senolytic purpose, Sport for high performance and faster recover, and T-Defense for anti-tumoral purpose. And this is the axis, this is the backbone algorithm. We use adaptive agent of neuroimmune, liposomal matrix, immune modulator, symbiotics, redox bioactives, and long-chain lipids. PLPC is the first, maybe the only, maybe the last nutraceutical with a validation ex vivo, because we work with STIP. So this is the profile that we get when, in ex vivo validation with any of the lines. And the final summary, it's a traceable cellular metabolic effectiveness with strategic modulation. It's not the same benefit. It's different in any patient, and some moleculars increase and some moleculars decrease. So you get the real profile that the patient needed. Best comparative position over the supplements, over nutraceuticals, over functional foods, and with full globally regulatory compliance. So at the end, what is the entry to this platform asset? It's very clean because the legal due diligence is pre-aligned with important legal firms across the world. The financial is very clean because it's through new companies for each asset in this platform. And scientific due diligence is open access. It's already done, published in papers and books. So at this moment, I think that maybe in the past, many investors or companies that develop a product or service like that committed far more for far less on the table, you know. But PLPC is different because it's not a concept, it's a service, a product, a platform with 11 years of history. And I think that is the unique moment to lead the next area on global immunotherapy, but first in class in structural immunotherapy. Thanks so much. Very well. Okay. Sure.
Thanks, it's a detailed presentation. I have a small question with reference to cancers like glioblastoma. As you're aware that it will grow in more immunosuppressive environments like brain. So how successful is this in terms of dealing such types of cancers? Number one. And the follow-up question is like, basically any cancer survives on 2 basic things. One is protein and other is sugar. Will this also impact the pathways of that environment where the cells get starved?
Okay, the first question is glioblastoma and central nervous tumor. We have many results and very successful. Why is the difference with PLPC? That PLPC is molecules, so the hematoencephalic barrier is not a problem if you use cells. This is one first. Second, the mechanistic about PLPC is reprogramming the microenvironment of tumor. So this is very benefit when you have a patient after radiotherapy, after surgery, after chemotherapy, with edema, with inflammation. In